continue, and I would like to ask you now to do the short reporting. We shorten it all down a little bit. We begin again with group 1, Bronwyn.
So we had a question from Diana about the different communities that you have there, and also the sort of what sort of genomes are we finding in these bacteria that are converting the ammonia, and as far as the work has been done, and I hope that Nathan will correct me here, they don't yet find the nitrophiles that are doing this conversion, and it's puzzling about the pathway that it might be taking. Is it as yet an unknown one? He talked of some interesting work done by Nancy Reelan on the jelly moonfish, and looking at the microplasma 16S sequencing. It might be a type of microplasma inside the cells that's doing this process. Then also questions on the effect of the nitrate being produced and then changing the ratio of nutrients, which will affect which species of phytoplankton will thrive on that and possibly support blooms.
Nathan, would you like to respond to Volker's question?
Yes, please. Thank you.
Could you tell me what was your question?
Yeah, so the question of Volker is a really good one. And of course, so I've shown that my results suggest that jellyfish blooms can be hotspots of nitrate and nitrite. And I've shown that in a specific place and time of that data, then it can supply between 2 or 4 times what is required for the primary production. And of course, the question is that, okay, but what is it on the whole coast, on the entire coast? Is it relevant? Is it an important source of nitrogen, like large scale and over the year? Of course, we could try to answer that question with my equation if we had a time series and good data of the jellyfish blooms density over the year. No, unfortunately, it's just recently that we start using sonar technology to quantify the jellyfish blooms, and the two maps that I've shown are the two best data, and that's just a snapshot of one bloom. But if we can have a much better data and time series on the blooms, then we can try to estimate how much nitrogen in general and the different forms of nitrogen will be released by blooms, and try to estimate how is that relevant for the productivity of the ecosystem or the primary production.
Okay. Then we go to group 2, Jose Luis. What would you have to report?
Well, mainly we have been discussing regarding especially 2 of the preliminary conclusions of the work regarding the relationship between the appearance of blooms and the effects of cyanobacteria and the concentration of ammonia and nitrogen, how it decreases as a consequence, when the cyanobacteria are acting, and how it increases when the bloom is coming, especially near the coast. The other questions were addressed regarding, regarding the amount of potential sources of nutrients to be considered in a coastal environment like this, near Mombasa, because it is necessary. This work is mainly focused on the effects, on the anthropogenic effects, and how that could be transformed with time after the application of new agriculture industries in the area. And finally, we've been from a methodological point of view. We've been dealing with the use of carbon and nitrogen delta in order to discriminate the potential sources of these nutrients in the area. Finally, we have a couple of questions from some Kurt Hanselman regarding how these ratios of bloom low nitrogen and bloom high ammonia and cyanobacteria low nitrogen could be extrapolated off the coast.
Okay, thank you, Luis. Nancy, would you have to add something?
Maybe just to say that I didn't say this, but like my work is conceptualized under it was motivated by the government. We have the Kenyan government implementation of new policy under the Big Four. We call them the Big Four agenda, where the government is going to implement to ensure food security through agriculture. And this will also lead increase in fisheries, and we are going to have more plantations. And this is what motivated my work. What will happen in case we increase agriculture and we increase industrial development without establishing appropriate waste management, organic waste management system? So this is what is making my work.
And I would like to go to group 3. Heidi?
Yeah, so we talked about how these crystals of purine or guanine could be formed. And Jana showed us some of her concepts that she already developed. And one of the key things, of course, that you have to know is that there's a membrane around them, a vesicle. And so the purine can be pumped into it. And it's actually more difficult to see how they could dissolve again, because they only dissolve at high and low pH. She explained that to us. And then we talked a bit about the connection to polyphosphate, as Jake was also there, of course. And very interesting that Jana observed that sometimes under stressful conditions, the algae won't divide any longer, but rather store guanine, and I'm not sure whether it's guanine or purine. I'm mixing those up a little bit. Sorry, Jana. And polyphosphate instead of dividing, which would then be the basic things that you need when you want to make DNA or RNA again. And then in the end, we had a little bit of a discussion on, or Diana explained us how in the early times they identified these crystals, and that was a bit by luck, actually, by feeding the right things to yeast, because it's a long time ago that these crystals were found. And then you kicked us out, unfortunately.
I'm sorry for that, but Diana, you may still clarify whether guanine is a purine or a pyrimidine.
Yeah, guanine is purine, and it's not easy to be precise all the time, because in most cases I observe guanine crystals, but to speak on general terms, I would rather speak about purine crystals.
Okay.
Because
obviously biochemistry is not my strong side, I have to say.
Okay, thank you. Any other questions? It's a fascinating topic, actually, how little cells can store, in this case now guanine, maybe as a guanine source, not so much as a nitrogen source or as both. But it's certainly a way to store nitrogen at all.
I can clarify also on this. We have tested it, and I would love to test it more and dig deeper. But for now, even with the preliminary experiments, we are sure that it's a common storage for nitrogen, because not feeding algae with any source of nitrogen, once they have only the inner sources of guanine crystals, they can divide rapidly. So they have to produce everything, even proteins, out of the guanine crystals.
Okay, thank you, Jana. Then we move to group 4 and Anya.
Actually, it was only the two presenters and myself. So I just had some methodological questions and asked about the value of the regression of the two species of the invasive semimetallus to the other species. And it's very clear that it's very early stages in the research. And then I also shared with them that I just learned from a colleague in the south of Namibia that it looks like semi-metallus is actually taking over the rocky shores in the diamond area in the south and moving further north. We didn't find any semi-metallus in Lüderitz when we were there in 2019, and we were supposed to be there this week, but due to COVID, we had to cancel it. So there's definitely some movement of semi-metallus, and there's definitely there are definitely more questions than answers. So when Kathleen asked if it would be worth doing a PhD on that topic, I said definitely yes, because as I said, we're in the tidal area. It's a very understudied and underreported area in Namibia. I don't know if Kathleen or Angelique want to add something.
To me, it didn't sound as a bad thing, because it might disturb the food web, but apparently it's not anyway disturbing the feeding of other animals. Is that correct?
Well, she said that because if rock lobster don't feed on them, it's a problem in the south, because that's rock lobster country if they're taking over.
Yeah.
Who is feeding on them?
Rock lobster.
Rock lobster.
Also on the invaded species?
Yeah, yeah.
No, they're not.
Yeah, especially the smaller one, the juveniles. They feed on them because they prefer like a smaller size. When I say, but we assume, and then also some of the gulls also feeding on them. So, but I don't have lots of knowledge, a lot of data on this. Instead, it's just like a very good interest now, and I want to explore and go more into detail. So I don't have the answers, only have questions. But when I look at the regression, it shows like a positive correlation, and that's why I thought maybe it's a good thing that there's all of a sudden abundant food resource. I don't know. So,
well,
but it's just,
okay. Well, any good research starts with good questions. So keep going, keep going. So we go to the last group. Paul, do you have to report something?
Quickly, we were a small group also of 5. Each one had to ask, each of the 3 had to ask a question. The first one there by Sarah was more a statement that the result seems to be a bit biased towards bottom trawling areas, and whether there was any chance of doing comparison with non-trawlable areas. And the response was given that this was like kind of a first trial to see what is there actually on our bottom grounds, because this was never done at this kind of intensive in the past. So it was kind of a first trial to see, and then design a new program to look at all that. Second question by Margit was, she was asking whether it was worth sampling this way, other than just trawls, or if whether the ministry has got a wider program to continue sampling around along the coast, especially at the mining sites. We discussed that we do know that diamond minings in the south, they do some kind of data collection. We have to find a way to get hold of that data, because the ministry actually now has to do it. There is no more, due to the Marine Stewardship Council certification of the hake fishery, which was achieved in November last year. The ministry has to design a program now to monitor the benthos and do the assessment. Last question was whether there is any environmental implications or effect of bottom trawling on the benthos. Damona answered that yes, and that was just when you were getting called back. I don't know whether she wants to extend on that, but I think the answer was given. Thank you.
Okay, thank you. Damona, would you like to add something or clarify?
About the mitigation measures, specifically on benthic invertebrates, I don't think there is much that is being done with regards to the, I mean, it's just bycatch that is in the trawls, and benthic invertebrates are those organisms that have less economical value, so there isn't much that can be done for the invertebrates. So obviously they get to be discarded after. So it's just package. The only mitigation, yeah. So yeah.
Well, I'd like to congratulate you, Damona, for your work and for the courage to do it. Okay, thank you for coming in person. Right, Sarah, would you like to add or make a suggestion to how they should continue that project?
Well, Margaret actually, she also asked like if, since they're taking samples by trawls, if they could actually take by sediment cores and see. And well, as Ndambona answered that this was opportunistic sampling with the fishermen, well, with the trawling. And this kind of monitoring would be amazing, but there's still no projects seen in the future. And yes, what I would like to add is that as Ndambona, as you put a figure there, that there's a really close link between the impacts of bottom trawling also on fish assemblages and benthic meiofauna, but it's also very closely linked to the sediment composition. So in order to really understand if what are the impacts on the ecosystem, we need to really do these interdisciplinary studies and hopefully using sediment cores that we don't disturb the seafloor, etc. But I don't know, hopefully there will be future opportunities to do this.
Yeah, Sarah, you are at the place where this can be done.
Yeah, okay. Then I'll try to.
Okay. Okay, anybody, any question?